Welcome to this video. In today's video, we're going to talk about virus. December 2019, a mysterious virus emerges in Wuhan, China. Within weeks, it spreads across continents, turning bustling cities into ghost towns. Hospitals overflow, millions of people fall ill, borders close, the world comes to a standstill. COVID-19 wasn't just a virus, it was a global crisis. It caused business to shut down, schools emptied, families separated, and over 6 million lives lost. It changed the way we lived, worked, and connected. But what if I told you this wasn't the first time? Viruses have shaped human history for centuries. The 1918 Spanish flu, one of the deadliest pandemics ever, affected 1/3 of the world's population and took over 50 million lives. Streets were silent, hospitals overwhelmed, and fear spread faster than the virus itself. Then came polio, which paralyzed thousands of children worldwide. Parents lived in terror until a medical breakthrough, the polio vaccine, turned the tide, which proved that science could fight back against those deadly viruses. The HIV epidemic in the 1980s brought a different kind of battle. A virus that didn't just attack immune system, it fueled stigma and fear. Decades later, antiretroviral treatment turned HIV from a death sentence into a manageable condition. But what exactly are these invisible invaders? How do they work? And why do some viruses cause mild colds while others trigger global pandemics? First, let's understand the virus structure. It's really important and interesting to mention that viruses are not cells. They do not have metabolism system, they don't have cytoplasm membrane or any functional organelles. Viruses are infection agents much simpler than any cell. Unlike bacteria, they cannot grow, eat, or reproduce on their own. They survive only by hijacking host cells to multiply. Because of this, they're known as obligate intracellular parasites. Despite their simplicity, viruses challenge our very definition of life. They do not metabolize, but they evolve. They do not grow, but they reproduce. They exist in the gray space between chemistry and biology, raise an even deeper question. Are viruses a remainder of ancient life, or are they nature's most efficient parasites? A typical virus consists of 3 main parts. First, the viral genome, which includes genetic materials. The virus blueprint, either DNA or RNA, contain the instruction to hijack host cells and make copies of itself. So, in the viral genome, they have either double or single-stranded RNA or double or single-stranded DNA, and also they can have linear or circular structures. Next, they have capsid. It is a protective protein shell that encases the genetic material, which is built from subunits called capsomeres that self-assembles into precise geometric shapes, from helix structure to isohedral symmetry. Next, they have envelope, which is an optional structure. Some viruses, like influenza and HIV viruses, have a lipid envelope stolen from the host cells. This envelope contains embedded proteins that allow virus to recognize and bind to new host, and this structure will make the infection process more effective. One of the important examples of viruses is bacteriophage. Bacteriophage is a virus that will invade the bacterial cell. It has a unique structure as the slide shows above. So on the left side, it is an example of T4 bacteriophage. It has a head, which is also a capsid. It contains the genetic material, usually DNA. Then it has a tail sheath. It has function like a syringe to inject viral DNA directly into the bacteria. Then it has tail fibers to help the phage recognize and attach to specific bacterial cells. Once attached to a bacterium, a bacteriophage can replicate using one of the two methods. The first one is the lytic cycle. The phage injects its DNA and hijacks the bacterial machinery to produce new phages. Eventually, it causes the bacterium to burst. The second option is the lysogenic cycle. The viral DNA integrates into the bacterial genome and remains dormant until triggered by outside conditions, and at that point, it will switch to the lytic cycle to replicate itself. Next, we are going to talk about the replication process of virus. First, I will introduce the lytic cycle. We have 5 steps of virus to replicate themselves: attachment, viral entry, replication, assembly, and release. The first step is attachment. Viruses recognize and bind to specific receptors on the host cell surface. It's much like a key fitting into a lock. This specificity determines which cell a virus can infect. The second step is entry. The virus can depend on its type. It may fuse directly with the cell membrane or be engulfed through a process called endocytosis. Release its genetic material into the cell's interior. Inside the cell, the virus commands the host machinery. The viral genetic material takes over, directing the cell to produce copies of viral proteins and nucleic acids. These components then self-assemble into new viral particles. Then release. New viruses are released. Often, this involves the host cell bursting open, which is a process known as lysis, which spreads the virus to infect neighborhood cells. In some other cases, virus exit the cell in a less destructive manner, cloaked in a portion of the host membrane. We can also see this process through an animation. So first, the virus come close up and bind to a specific receptor on the host cell surface. It then enters the cell either by fusing with membrane or being engulfed by endocytosis. The host cell's machinery is hijacked at this point to synthesize viral components, with this animated sequence showing that the production of viral proteins and genetic material. Then the host cell bursts open in a dramatic but controlled way, release a swarm of newly formed virus into the surrounding environment. And this process is called lysis. The second way of replication is the lysogenic cycle. Just like in the lytic cycle, the virus attached to a susceptible host cell and injects its genetic material. However, instead of immediately taking over the cell's machinery, the viral genome takes a different route. First, the viral DNA integrates into the host cell's chromosome and becomes what is known as a prophage. In this state, the virus is essentially camouflaged as it replicates passively along the host DNA every time the cell divides. During the dormancy, the viral genome does not produce new viral particle. It remains dormant within the host cell, sometimes for years, without causing any immediate harm or detection by the host immune system. This dormancy provides a survival advantage in those unfavorable conditions. The next step is induction. Under certain environment triggers, such as stress, UV light, or chemical signals, the dormant viral genome can be activated. This process prompts the viral DNA to exercise itself from the host genome. Once induced, the virus exits the lysogenic phase and enters the lytic cycle. It begins to command the host cell to produce new virus finally leading to the lysis of the whole cell and the release. After learning the replication cycle of viruses, it is important to learn the potential application of antivirus. In this case, stopping viruses in their tracks of replication cycle is exactly what antiviral drugs do. They target key stages in the viral replication to stop infection. Here are some examples. The HIV protease inhibitors prevent the virus from maturing and spreading. The influenza inhibitors can stop new viral particles from escaping affected cells. And COVID inhibitors can prevent the virus from copying its genetic material. The vaccine innovation. Some modern vaccines such as mRNA vaccines, which we discuss in class, can minimize the parts of the viral replication to train the immune system without causing disease. The mRNA COVID-19 vaccines will decrease the viral replication by instructing your cell to produce harmless viral proteins and trigger immunity. The influenza vaccines use inactivated virus, while new recombinant virus vaccines avoid using live virus altogether to enhance its safety. So what if we could use viruses to fix genetic disease? That's exactly how gene therapy does. Here are two examples. So these viruses can safely deliver corrected genes into human cells to treat those conditions. And scientists explore viral vectors to deliver function genes to patients with those illnesses. At the end of this video, I would like to express my deepest gratitude to Professor Lee for valuable guidance, amazing lectures, and support through this course. Your passion for biology has truly inspired me to explore deeper to this subject. A special thank you to our lab manager and technician, Dr. Li and Wang, for support and contributions to our lab session. Lastly, to my amazing classmates, thank you for the collaboration, discussions, and shared passion through this journey to biology. Thank you again for making this course an unforgettable experience. Thank you for watching.